Through cellular respiration, plants generate the energy they need. Carbohydrates, or sugars, are broken down and converted into energy by glycolysis, the Krebs cycle, and the electron transport chain. These processes, which are involved in aerobic respiration, generate a total of 36 ATP molecules. In this experiment, respirometers will be used to measure the respiration of different types of pea seeds. A solution of potassium hydroxide will indicate how much carbon dioxide is released by the peas during respiration. The first question for the pre-lab is, during respiration, a seed metabolizes sugars. What is the source of the sugar metabolized by the seed? When the seed is growing, it is not in contact with sunlight, so it cannot use photosynthesis to create sugars. Therefore, the seeds have to depend on organic compounds such as carbohydrates, proteins, and fats. And from those compounds, they obtain the sugar. And question number 2 is, what variables do you think affect the respiration rate of seeds? Brainstorm within your group to generate a list. And we said that temperature, the wetness or the dryness of the environment in which the seeds are placed, and the maturity of the seeds can be some of the factors that affect the respiration. In this part of the experiment, we started off with a water tray, and at the bottom of it we had a sheet of paper so that we could read the measure of the respirometers. In order to prepare the respirometers, we placed cotton balls at the bottom of each vial. As you can see in the diagram, there were 2 cotton balls that were used. One of them was soaked with potassium hydroxide solution, and the other one was used to prevent the contact of potassium hydroxide with the peas. And in the first vial, we placed 25 germinating peas. In the second vial, we had 25 dry peas as well as beads. And in the third vial, we had 25 beads. And we placed these respirometers in the tray for 5 minutes without submerging the tips of the respirometers. And after this time period, we submerged the tips, and every 5 minutes for 20 minutes we took the readings of the volume of air in each of the 3 respirometers, and we recorded our results in a table. For the second part of the experiment, our question was, how does water affect the respiration of peas? Our hypothesis was, if germinating peas are submerged in more water, then they will respire less most of the time. We concluded that this would happen because the water might diffuse into the pea seeds, thus reducing the amount of respiration that could be carried out by the cells. There are different variables involved in this lab activity, and an independent variable is the amount of water placed in each vial. The dependent variable is a measure of each respirometer, and the control variable is time. In this investigation, we had a water tray with a paper sheet at the bottom, and we also had 3 respirometers. One of them contained 20 milliliters of water, another one contained 10 milliliters, and the final one contained no water at all. 25 germinating pea seeds were placed in each of the respirometers. a timer was used also for 12 minutes at intervals of 2 minutes. We would take the measurement of each respirometer and record it. Here's a table of the results that we obtained. As you can see, the respirometer containing no water displayed a respiration rate of 7.8 at 2 minutes and also at 4 minutes. At 6 minutes, it dropped to 6.7. At 8 minutes, it decreased to 6.3, and it kept on falling to 5.9 and then to 5.4 at the end of the time period. And for the respirometer containing 10 milliliters of water, the rate of respiration remained at 8.5 throughout the entire time. For the respirometer containing 20 milliliters of water, the respiration rate was approximately 9.0. The results did support our hypothesis. We had predicted that the peas that were submerged in more water would respire less, and the measurements did prove that our hypothesis was correct. The respirometer containing no water, it displayed a measurement that decreased every 2 minutes, and the measurement indicated that the gas inside the tip of the respirometer was decreasing, and therefore respiration was taking place because there was more water entering the respirometer. In the case of the respirometer containing 10 milliliters, there was no decrease in the measurement because the amount of gas remained constant inside the respirometer. And in the respirometer containing 20 milliliters of water, there was hardly any respiration occurring. The respirometer was mostly filled with gas and did not allow water to enter.